Hello, everyone, and welcome to today's webinar, Enabling High-Throughput Biology with New Technologies for Synthetic Construct Sequencing. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by seqWell. To learn more about our sponsor, please visit seqWell.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd now like to welcome our speakers, Joe Mellor, co-founder and chief scientific officer at seqWell, Henry Chan, synthetic biology lead at Octant, and Bryan Jiang, research associate in synthetic biology technology at Octant. Joe, Henry, Bryan, welcome. You may now begin your presentation.
Thank you very much for that introduction, and welcome everybody to today's webinar. You'll hear today about how to enable high-throughput biology with new technologies for synthetic construct sequencing, which is an application webinar based on seqWell's new ExpressPlex technology. To tell you a little bit about seqWell, we really focus on how to solve some of the crucial bottlenecks in NGS workflows. Our goal is to help people sequence more samples on sequencing instruments more easily, and we do so with workflows and products that are designed to provide enhanced multiplexing, allowing you to take samples and prepare them into NGS libraries and get them onto sequencers more quickly, more easily, with higher performance. A crucial technology that we use to build almost all of our products is an enzyme called the TN5 transposase, which is an incredibly versatile tool for scaling and simplifying NGS workflows. We have found TN5 to be an incredibly powerful platform for creating library chemistry with a number of unique advantages. For example, being able to normalize and create multiplexing with purpose-built reagents. These reagents really form the core capabilities that our products are based on, and we also have a lot of experience in creating products that support specific applications, for example, like plasmid sequencing, which you'll hear about today. We think of these products as really the product of a workflow engineering approach that we have created that builds on the core reagents themselves and then creates streamlined products that deliver cutting-edge integrated workflow. Many of these workflows are built into other products that seqWell has offered and continues to offer, starting with our PlexWell product and most recently culminating with our PurePlex product launched last year and ExpressPlex, which you'll hear about today. All of these products really form the basis of a series of different technologies that support a range of different multiplexing needs across different applications such as genotyping, RNA-seq, genome sequencing, and plasmid and amplicon sequencing. ExpressPlex is a very exciting new product that we're launching this year, which you're going to hear about in today's webinar. And this product is really the culmination of work on the part of our R&D team to create the fastest, most highly automatable library prep possible. Think of ExpressPlex as being the simplest possible way of turning DNA into a sequencer-ready library. The way this works is we create plate-based products that allow you to put the DNA into a single reaction mixture and generate a high-performance library from hundreds or even thousands of samples quickly and easily. All of this can be done in a total time of 90 minutes from the DNA sample itself to a sequencer-ready library. As you'll learn about today, this has a number of applications, and you're going to hear today about the application of plasmid sequencing that our collaborators at Octant have been exploring. And so with that, I'm going to turn this over to our collaborators, Henry. Henry Chan is the synthetic biology lead at Octant, and Bryan Jiang is the synthetic biology research associate. They're going to tell us today about their work in deploying ExpressPlex as part of the OCTOPUS protocol, which they have developed to support high-scale, high-throughput plasmid sequencing. Henry and Bryan, it's all yours.
Awesome. Thanks, Joe. Yeah, so we're really excited to be talking to you guys today about this joint collaboration between us and seqWell. Real quick, I'm going to give a quick introduction on Octant. We are a we are building a platform to navigate the complexity of biology and chemistry to develop the next generation of small molecules. So we're a therapeutics company. But more specifically today, I want to talk a little bit about our efforts with seqWell to help us develop and improve OCTOPUS. This is a high-throughput construct sequencing platform that we've used that really helps enable our drug discovery efforts.
And we're excited to share with you all today on how seqWell's ExpressPlex kit has enabled the possibility for the next day turnaround of full plasmid sequence verification using Octant's OCTOPUS v3 platform that we'll talk later about. We shaped this upcoming OCTOPUS update so that any interested lab or party can implement this within their own workflows. Henry is going to give some context for how onboarding ExpressPlex onto OCTOPUS has enhanced our company's synthetic biology workflow and overall platform.
Thanks, Brian. So really quick, yeah, so we are a drug discovery company. Our fundamental hypothesis is that the current state of drug discovery is poorly matched to address the multifactorial nature of human disease. We think that next generation therapies are possible through understanding the right biological mechanisms that target the right receptors, the right pathways, and the right mutations, right? Disease is complicated. We don't believe that we don't believe in the silver bullet hypothesis. What do I mean by this? So let me give you an example. So autosomal dominant retinitis pigmentosa is a disease that leads to progressive blindness. It is often caused by misfolding mutations in the rhodopsin gene. This leads to death of retinal cells. And the mechanism behind how this is, if you look over on the figure in the middle, misfolding mutations of the rhodopsin protein will lead to mistrafficking of the protein from ER, where it is created, to the plasma membrane. Small molecule chaperones could be a potential therapeutic for this. And so you could imagine a molecule that might bind to the misfolded rhodopsin protein and then help it adopt a more native and wild-type conformation. This adoption of the conformation will rescue the trafficking and then prevent the cell death. And so you might think one way to find molecules that could have this chaperoning activity is you might purify wild-type rhodopsin and then look for small molecule binders or stabilizers of the wild-type protein. And you might, that is certainly a fair way to address this problem, to look for, to screen for interesting compounds with therapeutic effect. But we believe that if you take an approach like this, you're definitely leaving a lot of potential mechanism on the table. And so when in the process of, in the cell, right, a misfolded rhodopsin protein will undergo all these different processes, will experience all these different processes, right? Like biogenesis, translation from the ribosome, trafficking from the ER to the Golgi to the cell membrane. The molecule will have a potential therapeutic or chaperone will have many opportunities to interact at all of those complex stages of trafficking and folding. Additionally, there's not just one single kind of mutation, right, that leads to adRP. There are actually hundreds of known mutations. And so a truly best-in-class therapeutic you might want to address many or most of these mutations, and you don't want to leave any mechanism on the table by only screening for binding to wild-type protein. You want to look for things that affect the biogenesis and trafficking at the cellular level. And so this is one of our fundamental hypotheses, right? It is around this hypothesis that we've built our drug discovery platform, which we're calling the Navigator. To that end, we have two kind of platforms. The biology platform, where we utilize multiplexed cellular assays to help us optimize drugs and to do more precise drug design to multifactorial mechanisms and mutations. And then the other side of this, the other part of our platform, which I won't talk about today, is our high-throughput chemistry platform, which allows us to design and iterate and generate tens of thousands of molecules every week. And this, rapid iteration allows us to really quickly optimize our drug designs more rapidly. And so we refer to our biology side as cellular intelligence. And this is based a lot on technology that our founder, Sri Kasuri, has pioneered. So Sri did a lot of research as a professor in synthetic biology and genomic technologies, really pioneered multiplexed assays and these types of technologies like synthesis, gene editing. And so a lot of our biology platform is built around our ability to engineer human cell lines with genetic barcodes. These barcodes are able to report on the activity of different targets in multiplex. And when we build these multiplex barcoded assays, we use these as screens and assays for identifying and optimizing interesting therapeutic compounds. And so the two main ways in which our multiplexing technology breaks out are in the broad target scan and in the deep mutational scan, BTS or DMS. So in the BTS, we can screen 100 different targets or 100 different mutations of the same target, or a mixture of both, right? In 384-well format. And at this scale, we can do tens of thousands of molecules against 100 different targets or variants or mutations. The other side of this is a deep mutational scan. Here we can screen tens of thousands of mutations in parallel. So you can imagine we can make every single amino acid mutation to a given protein and then map out all of that at once. In a big dish. And so this could get us to understanding some SAR or structure activity functional relationships at a very granular level, whereas on the BTS gets us these broad chemical screens at many targets more broadly. How did this play out for adRP? So we built a multiplex trafficking assay, and as I told you before, rhodopsin has hundreds of known mutations that cause retinitis pigmentosa. We were able to build many of these and then multiplex them into a single high-throughput screenable assay. And then from that, we were able to find compounds that actually rescue 80% of the trafficking mutations that we tested. So in this plot on the bottom right, you can see we've identified 3 compounds that have this level of rescue. Each square is actually a dose response curve. And because we're multiplexing, the density of data that we get is actually quite high. So in a 384-well plate, we're getting like hundreds of dose response, equivalents of dose response curves that you might collect on, say, like a luciferase or fluorescence assay. But this is against many variants at the same time. Building these assays, however, requires building and developing these assays requires a lot of work. Tons of cloning optimization were involved, and so in building the assay that we just talked about, we optimized at least 14 different synthetic components to be able to achieve a signal that we were satisfied with. And in this process, we tested at least 400 constructs. That is not the end or the only multiplex trafficking assay that we want to build. There are many different mechanisms that we want to pursue. There's an example, it's not just some example of the ones on the left that we can build in cell assays. And then there's also the reporter element that expresses the barcode. And so we've also found a lot of things to optimize both on the mechanism side and the promoter side that actually expresses the barcode. And so in going after building these multiplex assays, we actually go through quite a lot of iterations of designs and cloning and cell engineering. And so rather quickly, we had to get really, really good at cloning. And so we adopted, a lot of the latest gene synthesis. We take full advantage of a lot of latest gene synthesis technologies and the pooled cloning approaches. But what we really ran into really quickly was that something as plasmid sequencing, sequence verification by Sanger became a huge bottleneck. We were sending in thousands of plasmid traces a week to pretty large CRO. We were incubating out of UCLA, and so I won't name who the CRO was, but we were spending quite a bit of money on them, and they were throttling us at 90, at 2 to 4 96-well plates a day. This meant that we were having to wait a week to get our data back, not to mention the cost, and then also just submitting, tons of primers and analyzing thousands of traces. This was not fun at all. And so we asked ourselves the question, we're an NGS company, next generation sequencing company. We live in the age of NGS. Why are we being bottlenecked by decades old technology for DNA sequencing? And so the reality is that this is a solved problem. If the name of your company is Ginkgo or Amyris, and you've built your company around DNA around like this sort of synthetic biology. We were a startup. We had 2 molecular biologists that were doing all of this cloning. We obviously couldn't afford to build this world-class setup that they had. And this is, back in 2019, this is when we developed OCTOPUS. This is a cheap and scalable high-throughput full-plasmid sequencing platform that is super scalable, is direct from colonies, so no mini preps required. It's accessible, no fancy equipment. You only need a MySeq and maybe some multi-well pipetters. Fast turnaround is comparable to Sanger. Automated analysis report, you don't have to sit in front and stare at traces. It just spits out an Excel file. It's cheap, costs the same about a Sanger. And like any good automation and process, it changed the way that we approached experimentation. Things that we were able to do like on the bench side in the molecular biology lab, it just we were able to just take riskier and riskier cloning approaches and go crazy with what we wanted, because we had this thing on the back end that would just tell us what we were doing at a high scale. And then the other thing that we thought was we should open source this. And so you can actually find our protocols for exactly how to do this on GitHub. We wrote a blog post about it that's written down on the bottom. And so in a nutshell, really quick, how this process works. On day 1, you pick colonies. Day 2, get in really early in the morning, you lyse the cells, you extract the DNA in bulk at the plate level. Then you do the library prep. At the time, we were using the iGenomics RIPTIDE kit for library prep. That takes about 6 hours. And then by that evening, you could load your MySeq or your next-gen sequencer. And then by then around, less than 48 hours later, you get your results, your analyzed pipeline results. And so as you can see on the plot over on the top right, in the first 6 months that we developed this, we loved it. We basically sequenced 12,000 plasmids on this using OCTOPUS. And again, as I told you, we only had like 2 molecular biologists, and we had 1 RA that was running this. But there were still some things that were not perfect about it, and I'll pass it off to Bryan to tell you about it.
Yeah, thanks, Henry. Now, I wasn't around the time to run V1 of OCTOPUS, but from what I understand, running day 2 sample prep and library prep by hand was quite a pain in the butt. And also, the cell lysing method being used resulted in higher gDNA bias than we would have liked. So reducing the hands-on labor required and the gDNA bias culminated in the V2 update of OCTOPUS. One major change was the onboarding of rolling circle amplification to replace the older cell lysis sample prep method that we had. Modifying RCA with the addition of plasmid-specific primers greatly improved the quality of our sequencing by reducing the amount of gDNA contamination. Additionally, we utilized the Opentron's OT2 liquid handling robot. This robot was cheap automation that really further reduced the amount of hands-on laborious work required to get through the day 2 library prep. But while we addressed the gDNA and hands-on labor problems with the V2 update, the library prep process itself, while still using the library kit that we had, still had a lot of moving parts that made the process more complicated than we would have liked.
Yeah, and so then this is actually around last year. This is when we got in contact with the folks over at seqWell. Roger and Joe, they found us through our blog post and they reached out to us and they said, hey, we have this kit called ExpressPlex we think you might be interested in. And they showed us this protocol, doesn't get simpler than that and takes 90 minutes. Compares that to the RIPTIDE protocol, which if you guys know, RIPTIDE was bought out by Twist. It's now called Twist 96plex, but we refer to it as RIPTIDE because we actually never bought any kits from Twist. We had so many of the RIPTIDE kits. But anyway, just compare that to the RIPTIDE protocol. Super long, takes like about 5 to 6 hours, many steps, many ways that you can mess this up. There was always training involved. And so we saw this protocol, the ExpressPlex protocol, and we were like, yes, we would love to be the alpha testers for this product. And so we brought it on, and our early prototyping, our kind of early testing was we found out that the sequencing bias between RIPTIDE and ExpressPlex was basically the same in terms of performance. If not, maybe ExpressPlex even has a slight edge on it. What I'm showing you here is just like kind of bias plots from 2 random plasmids that we've done. We've done more rigorous statistical analysis on the back end to convince ourselves, but I think the takeaway is, to date, we've sequenced now 65,000 plasmids using the RIPTIDE library prep kit. Never experienced any issues. Obviously, bias is going to be introduced whenever you have library prep. It was fine for us. And so when we saw something that was basically just as good, if not better, we were super happy with that. And as of today, we sequenced around 7,000 to 10,000 plasmids using ExpressPlex library prep kit, and again, no issues there. And so with that, we were really excited. Actually, Bryan was really excited to bring ExpressPlex on board.
Yeah. Basically, with the simplified and faster library prep that ExpressPlex allowed, it made my life a lot easier. And running OCTOPUS, and it shortened the library prep day quite a lot. But this also got us thinking that if we were able to make the library prep day this much shorter and able to turn RCA that was formerly a overnight process and squeeze it into the same day as the library prep, we might be able to get next day turnaround of our plasmid sequencing. And indeed, we'll talk about this later, but yeah, this culminated, with our upcoming V3 update of OCTOPUS by combining the speed of and simplicity of the ExpressPlex library prep with a shorter RCA protocol. We were able to achieve next day turnaround of full plasmid verified sequences. And additionally, the library prep process can be assisted by the OT2.
Yeah, I mean, this is crazy, right? Like, it's literally faster than Sanger. And as Bryan mentioned, we're going to be releasing these protocols soon. Just be on the watch out for that. We haven't actually written everything up yet, but it works. It's awesome. And so this, again, like this changes everything for us. It's basically taking a 2-day process to 1 day. This dramatically shortens time to data, right? And so in our workflows, you can imagine, if you order, say you're super excited from, coming in from thinking about your problem on the weekend, you come in on Monday, fired up, you're ordering primers, you clone them, and then IDT delivers them on Tuesday because they're amazing. And then you clone on Tuesday, and then you do OCTOPUS on Wednesday. You get your results by Thursday, and you can then now set up your experiment, or if you mess up, reorder primers on Thursday. And if you set up your experiment on Thursday, then you can, for us, we transfect on Friday. That allows you to take advantage of the weekend incubation or reclone on Friday. And so because of like the weekend, right, and the way that, the week breaks out and lab work just happens, this 1 day shortening of the process gives us 2 turns of the crank on any given experiment rather than 1. This is huge, right? The other side of this too is this easier workflow. Until now, everybody has had to rely on Bryan to run OCTOPUS for them because of this super long and involved library prep. Only Bryan knows how to make the OT2 sing like it does. But now it's just, 1-step transfer and then put it onto the library. We can actually democratize this within Octant and people can run it. Our vision is that anybody who does molecular biology can just run OCTOPUS whenever they need to and not have to wait for Bryan. The other side of this too is, a lot of people reach out to us about OCTOPUS, and I know of at least 10 companies that are probably using OCTOPUS right now. And I would imagine many more would use it if they knew it was this easy and was literally faster than Sanger. And so we also imagine, right, there are more turns of the crank, but also more turns of the crank democratized out to everyone within Octant, but also outside of Octant too, in which we're really excited to be able to share this protocol. And so in summary, Octant is building we are a drug discovery company. We're trying to build the next generation of small molecule therapeutics by leveraging synthetic biology and multiplex cell-based assays and our high-throughput chemistry platform. OCTOPUS has been this tool that we've developed has been greatly enabling for the synthetic biology efforts here in the engineering of cell-based assays. And with this collaboration and the advent of ExpressPlex into the workflow, this reduction in turnaround time, labor, and expertise for OCTOPUS has greatly increased the usefulness and accessibility of this.
Yeah, and at its fastest, you can have with the OCTOPUS input, you can have transformed colonies on day 1 to mini preppable verified culture by the next day. Our platform will be freely available for open source collaboration and mutual benefit across all labs, as well as an OT2 protocol to further simplify the wet lab process.
Yeah, and with that, I'd like to just some shout outs here. Nathan Lubach, he was a scientist, a bioinformatician at the time. He wrote most of the analysis pipeline for Octopus. Johnny was that first RA we talked about, made a lot of improvements to V1 and V2. Liz was a second RA runner. She made some improvements to the wet work. Will kind of spruced up the analysis pipeline for us a little bit. And then Bryan has done a lot of improvements to V2 and V3 and is the current runner for us right now. For the Rhodopsin project, Eric Jones is the lead scientist. And then our two founders are Shri Kusuri and Ramsey Hamsani. And then the folks over at seqWell, Joe, Roger, Curtis, Jack, Kelly, Samantha, you guys have all been great and super helpful in this collaboration. We're excited to continue working with them. And so with that, I'll thank you guys for your attention and give the floor to any questions.
Well, thank you, Joe, Henry, and Bryan for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Our first question asks, are the protocols for Octopus and ExpressPlex published or available to review?
Joe, I think the ExpressPlex protocols are literally like what you see in the slides. I don't know if you have more than that, but in terms of Octopus, we are currently trying to write up the protocols and formalize them. We hope to have that finished by the end of March to get a sense for like what the workflow is for Octopus. Sans the like fast RCA and the ExpressPlex. You can take a look at our GitHub and our blog posts. There'll be a much longer version of the protocol there, but just stay tuned. We're trying to write them up as we speak.
Yeah, and just to add to that on the ExpressPlex information, certainly anybody who's interested in learning more can reach out to any us at seqWell. This product will be officially launched next month, and so we're certainly looking forward to publishing a lot more information on protocols and et cetera on our website soon.
Great, thank you. Another question we have here. How much time did it take you to integrate ExpressPlex into your overall workflow?
So the ExpressPlex kit, it's as simplified, it's as simple as it can really be, just stamping and a single thermocycling reaction. For us, we were able to implement ExpressPlex within Octopus on the first attempt. In the first run, we were able to get sequence verified results. The kit does its job, right? It converts plasmid input into a Illumina sequenceable library, and the Octopus process, basically, that's just what it requires. It's just a faster, more simplified version of the system we had before.
Great. Next question here. What is the maximum number of samples, small plasmids, say 10 kb, that can be multiplexed within a single flow cell?
I can take that one. So I think it depends on the sequencing kit you use. We routinely use the V2 micro from the MySeq from Illumina. And so I think that gets you 4 million paired in 150 reads. And so very, very comfortably, that is 8 96-well plates. I'm pretty sure you could push that up to twice that at the 10 kb size. But I think the limit there for us is actually the number of unique indexes.
Great, thank you. Next question is a two-part question here, and this question asks, in your experience, how long does it take to prepare the sample normalization to start with the protocol? And the second part of the question says, can you please briefly explain how you use RCA?
Yeah, so I think I'll take the first part just to maybe address the normalization question. So I think one of the really attractive features of ExpressPlex is it does normalize over a pretty wide range of input DNA amount. So again, I think a typical use case certainly could be a global dilution, not necessarily an individual normalization of samples. I think, maybe, again, Bryan and Henry can speak a little bit to how the output of the RCA process kind of fits with that concept briefly.
Yeah. So the cool thing about the RCA process is when RCA is run to saturation, that also is a step of normalization of all the input samples. So on top of that, it requires it doesn't require like spot quanting at the beginning. After you have your sample and RCA is run to saturation, you can dilute that by a global factor. As you can expect all wells to have roughly the same amount of input. And then that can be stamped into the ExpressPlex kit.
Right. We have another question here. Can ExpressPlex be used for applications other than plasmid sequencing?
Yeah, so the answer is absolutely yes. And I think, certainly initially our focus applications will be in a wide variety of synthetic constructs, including things like amplicons. These are sort of key applications we see this being a great fit for now. And we will certainly look forward to expanding beyond that. I can tell you, certainly we've had some really positive data on other diverse applications such as RNA-seq that we're looking forward to telling the world about more quite soon. So again, I think the answer here is, it's a new product and we're excited to get it out there and certainly learn from our customers about where it can go. But I think initially, certainly applications like the one you see here today are going to be the ones that this is really well suited for.
Great, thank you, Joe. Our next question here asks, how many samples are you processing at one time or sequencing in a batch? And how many reads are required per sample?
So with the Illumina micro kit, we sequencing 150 base pair paired end, we comfortably sequence with just 8 indexes, 8 times 96 samples, around 800 samples, comfortably getting an average of around 5,000 reads per sample. That's plenty of coverage for a 10 kb plasmid input. With more indexes, it's possible to imagine running maybe up to 1,200 samples or 12 indices multiplied by 96 samples within a microkit. The Octopus platform, the analysis pipeline, is able to accommodate low those levels of read coverage because it doesn't rely entirely on de novo sequencing, which would require a lot of reads, but it has an intermediate step basically allowing for full plasmid sequencing upon with the low input sequencing kit.
Yeah, and then I think one day, if there is a need to do more than 896 whole plates per run, I think one that's totally possible with the micro kit to, the V3 kits are like 30 million reads. So you can imagine like 10 times the number of samples. Like if you are, one of these like big cores, or you want to, if you want to eat the lunch of these Sanger companies and try to conglomerate like, the entire country's worth of like plasmid sequencing in, all in a single location, you probably do that too. I think again, you just bottleneck by the number of unique indexes.
Yeah, and alternatively, if like those are like pretty large scales, you can also easily imagine that using a nano kit, would be sufficient with like just like 1 or 2 96 well plates worth of samples. That would probably also give you, 5,000-ish like reads per sample, and that's like plenty of coverage for 10 kb plasmid.
Great.
For OCTOPUS.
Now, Henry and sorry.
Sorry, yeah.
Great. Well, Henry and Bryan, I have another question coming your way, and this is another 2-part question here. This question asks, do you see this technology and approach replacing Sanger sequencing at workflow and costs that labs can change, especially core labs and high-volume Sanger CROs service providers? And the second part says, what would you pass on as words of wisdom from your experience to folks thinking about this change?
I think doing like a complete 100% replacement of Sanger is probably not like what this would do. But at the same time, probably close, like the vast majority of plasmid sequencing. I don't understand why that, like, I don't see why that wouldn't be the case. We still have a account, over at our CRO, but I would say like 95% of the plasmids or more even that we generate here at Octant are sequenced by OCTOPUS, and especially with the easier workflow and the shorter turnaround time, that is actually faster than what Sanger can actually provide. Like, there's no reason why, for most things that OCTOPUS can handle, you wouldn't do this. Like, I think as in the question, if you don't have high volume, like, I think it stops to become kind of, it's not as worth it at the sub 96 well level. I think like right around there, it's really worth it to do. But if you are a high volume lab, or you're like a core facility at a university or something like that, it just doesn't make sense to me that you would ever use Sanger. I think there are some things that one, we're not selling OCTOPUS, so like we try to develop it to the point that's useful to us, and we share it with the world. But there's probably some tweaks to the analysis pipeline that might make some things a little bit tighter. And then the other thing is, of course, like with any kind of sequencing library prep, there will be bias, if you have highly repetitive sequences littered throughout your plasmid, which there are all sorts of problems with that, just from the cloning and the design side. Anyway, like I think we've seen that OCTOPUS can choke on that a little bit. We can still sequence it, but it's not perfect. So every now and then we'll just like use Sanger to like spot check some things that seem kind of crazy. And then in terms of like words of wisdom, I think just email us. I like, honestly, I would have, if it wasn't this easy, like if it was still V2 or V1, I would have a lot more things to say. But when we spin up the protocols, definitely email us to see what might help. And yeah, I don't know. It's so easy. Like I don't see any. I don't anticipate any wisdom needing to be shared other than having to have this scale. Bryan, I don't know.
Yeah, pretty much. Once you're approaching the scale of 96 samples or more that need to be fully sequenced, or it doesn't have to be fully sequenced even. Like, it's because of how easy it is. If you're on the 90, like at that point, analyzing Sanger traces like becomes like the pain. And having OCTOPUS as an option, even as like a first pass option, right? You pretty much get everything you need. And if you're still like more comfortable with Sanger, you can easily Sanger. But over time, you'll well, at least like, for us, like we quickly realized, the octopus is like pretty sufficient for almost like all use cases. And especially for cases where you have, complex like plasmids, like, cloned in, like de novo, or or just having a lot of samples. And because all the input samples don't have to be mini prepped, you literally start from bacterial colonies. That like further simplifies the process. You don't have to like consider like, oh, I have to mini prep, like 8, like, maybe of like this particular construct. You can just like pick your colonies, put it in, and then just mini prep the single, like perfect at the end. So that's just another benefit to the Octopus process when you're approaching the 96 well scale.
Great. Well, looks like we have time for one more question here, so we're going to wrap up with this one. And this question asks, are you looking at any of the new NGS platforms?
Yeah, so I think, short answer there is, I think certainly from our point of view, multiplexing is a key need that spans, various sequencing platforms. Certainly, as newer platforms have come out, especially in the last year, I think we've seen that now become more and more apparent. At seqWell, we've done, I think, some really good work that I'm happy to direct you to our website and check out. We've shown really good performance with our some of our other kits with the Element platform, the Aviti sequencer. But certainly looking across, the spectrum at other platforms, I think there's some really exciting stuff going on, certainly in the long read arena. Some of the new platforms in that space, I think, offer throughput and cost that now really is going to motivate a high level of multiplexing as well. So yeah, we're very excited to start to think through how to implement some of these technologies on other sequencing platforms as they get out there. But yeah, more to come, and certainly I certainly invite you to take a look at, again, some of the work we've already put on our website in that regard.
Awesome. Well, thank you again, Joe, Henry, and Bryan, for your time today and your important research. We would also like to thank LabRoots and our sponsor, seqWell, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.